Water is one of the most essential components for plant growth and survival. In most herbaceous plants, water constitutes about 80–95% of the total fresh weight of the plant body. It acts as a solvent for mineral nutrients, participates in metabolic reactions such as photosynthesis, maintains cell turgidity, and serves as a medium for the transport of nutrients and organic compounds within the plant. Because plants continuously lose water through transpiration, they must absorb an equivalent amount from the soil to maintain their physiological balance. The process of water absorption mainly occurs through the root system, particularly through the root hairs that are specialized structures for increasing the surface area of the roots. Water movement in plants is governed by the concept of water potential, which determines the direction in which water moves within plant tissues. Water potential refers to the potential energy of water relative to pure water and indicates the tendency of water to move from one region to another. Water always moves from regions of higher water potential to regions of lower water potential. The water potential of a plant cell is influenced by several components, including solute potential, pressure potential, gravitational potential, and matrix potential. In most physiological studies, the major components considered are solute potential and pressure potential. The solute potential results from dissolved substances within the cell sap, while pressure potential represents the physical pressure exerted on the cell walls due to water entry. Water absorption takes place primarily in the region of the root where root hairs are present. Root hairs are thin-walled, tubular extensions of epidermal cells that greatly increase the surface area available for absorption. These structures come into close contact with soil particles and the thin film of water surrounding them. Water enters the root hairs mainly by the process of osmosis because the cell sap of root hairs usually has a lower water potential than the surrounding soil solution. As a result, water moves into the root cells across the semi-permeable plasma membrane. Once water enters the root hairs, it moves inward through different tissues of the root until it reaches the xylem vessels, which are responsible for transporting water throughout the plant. Water can move through the root cortex by three main pathways: the apoplastic pathway, the symplastic pathway, and the transmembrane pathway. In the apoplastic pathway, water travels through the cell walls and intercellular spaces without crossing cell membranes. This pathway allows relatively rapid movement of water. In the symplastic pathway, water moves through the cytoplasm of adjacent cells connected by plasmodesmata. The transmembrane pathway involves repeated crossing of cell membranes as water moves from one cell to another, often facilitated by specialized protein channels known as aquaporins. A crucial regulatory structure in water movement through the root is the endodermis. The endodermal cells contain a specialized structure known as the Casparian strip, which is a band of suberin deposited in the cell walls. This strip blocks the apoplastic pathway and forces water and dissolved minerals to pass through the plasma membrane of endodermal cells before entering the vascular tissues. This mechanism ensures that the plant can selectively control which substances enter the xylem, thereby protecting the plant from harmful substances present in the soil. One of the mechanisms involved in upward water movement in plants is root pressure. Root pressure is a positive hydrostatic pressure generated within the root system due to the active accumulation of mineral ions in the xylem vessels. When mineral ions are actively transported into the xylem, the solute concentration within the xylem increases, causing the water potential to decrease. As a result, water moves from surrounding root cells into the xylem by osmosis. The continuous influx of water generates a positive pressure that pushes water upward through the xylem vessels. Root pressure is particularly noticeable during periods when transpiration is low, such as during the night or early morning. The presence of root pressure can be demonstrated by several observable phenomena in plants. One of the most common examples is guttation, which is the exudation of water droplets from the tips or margins of leaves through specialized structures called hydathodes. Guttation usually occurs during the night when soil moisture is abundant and transpiration rates are low. Another evidence of root pressure is the exudation of sap from cut stems, often referred to as bleeding. In plants such as grapevines and sugarcane, sap can flow out of cut stems due to the positive pressure generated by the roots. Although root pressure contributes to the movement of water within plants, it is not strong enough to account for the transport of water to great heights in tall trees. Root pressure is relatively small and is not present in all plant species. Therefore, scientists have proposed another mechanism known as the cohesion tension theory to explain the long-distance transport of water in plants. This theory, proposed by Dixon and Joly in the late 19th century, states that water is pulled upward through the xylem due to the tension created by transpiration in the leaves. According to the cohesion tension theory, when water evaporates from the surfaces of mesophyll cells in the leaves during transpiration, it creates a negative pressure or tension within the leaf tissues. This tension pulls water from the xylem vessels in the leaves, which in turn pulls water upward from the stems and roots. Water molecules remain connected to one another through a property known as cohesion, which arises from hydrogen bonding between water molecules. At the same time, adhesion between water molecules and the walls of xylem vessels helps maintain the continuous column of water as it moves upward through the plant. Stem pressure and xylem transport are therefore largely influenced by the combined effects of transpiration pull, cohesion between water molecules, and adhesion to the walls of xylem vessels. Together, these forces create a continuous upward movement of water from the soil, through the roots and stem, and finally to the leaves where it is used in photosynthesis and other metabolic processes. Several factors influence the rate of water absorption in plants. External factors include soil moisture availability, soil aeration, soil temperature, and soil salinity. If soil water is limited or the soil becomes highly saline, the water potential of the soil decreases, making it more difficult for plants to absorb water. Internal factors, such as the size of the root system, the permeability of root cells, metabolic activity within the roots, and the rate of transpiration also play important roles in determining the efficiency of water uptake. Water absorption and upward transport in plants are complex physiological processes that involve coordinated interactions between roots, stems, and leaves. Water enters the plant primarily through root hairs by osmosis, moves through root tissues via apoplastic and symplastic pathways, and enters the xylem after passing through the regulatory barrier of the endodermis. Root pressure can contribute to the upward movement of water under certain conditions, but the primary driving force for long-distance water transport in plants is the transpiration pull generated in the leaves. As explained by the cohesion tension theory, understanding these mechanisms is essential for improving agricultural practices, managing irrigation systems, and developing crop varieties that can tolerate water stress and environmental challenges.